Okay, we are going to start Chapter 5, which is metabolism. Probably have 2 videos on this tough topic. You'll notice I have a variety of pictures here. I have some foods, drinks. I have plates, tubes, biochemical tests. And I got some guys cleaning up the beach up at the top. And I'm trying to show you the use of microorganisms in the environment. For example, top left is cleanup of oil in sand using Pseudomonas putida, which breaks down hydrocarbons, and oil, which is hydrocarbons. And it does it by metabolic pathway, having enzymes to do that particular pathway. But also microorganisms are used to make bread, make beer, ferment food of all sorts, including salami at the bottom right. And of course our biochemical tests are based on the same metabolic pathways. Okay, so think about metabolism. It's the same as metabolism you studied in general biology. It's all biochemical reactions in a cell. You can look at this list. It's right out of your textbook. We'll talk about this as it comes up as we get to the material. Okay, so you need to review some things that you know. 2 breakdown areas. 2 ways to look at metabolism. Building reactions, anabolic reactions, or pathways that do this are called anabolic pathways, or the whole process is called anabolism. Anabolism requires lots of ATP energy input. Catabolic reactions, catabolic pathways, or catabolism, are breakdown degradation reactions that produce large amounts of energy, or ATP. Now, all chemical reactions require some energy to get the ball rolling. There's got to be some ATP input, some energy input to get chemical reactions going. So there's energy required in varying amounts in both cases, but catabolic reactions produce far more, generate far more ATP than you need it. Okay, exergonic versus endergonic, energy producing, energy using, ATP, okay, blah, blah, blah. Okay, so let's get to oxidation reduction reactions. There are different ways to define oxidation reduction. You can define it from the standpoint of oxidizing a chemical by adding oxygen, or reducing a chemical by removing oxygen. Forget that. That's used in chemistry, but not the way we're going to use it here. You can talk about oxidation and reduction as being the addition of hydrogens, hydrogen ions. That would be reduction, or taking them away is oxidation. That's sometimes used in biology. But the third one I want to give you is the absolute definition used in biology. And that is the gain or loss of electrons. So oxidation is the loss, giving away electrons, and whatever molecule picks those electrons up has been reduced. That is reduction. So look at the picture down at the bottom right, and you see, let me go over to my pencil here. Okay. All right. So you see an electron right there, and it's going to get passed from that molecule, whatever it is. Maybe it's a glucose molecule. It gets passed to another molecule, NAD, sometimes just called NAD+. And when that electron is given to NAD, that's where it's going to end up. NAD now, which is now called NADH, right there, has been reduced. It has gained electrons. Now the guy that gave them away, this guy here, has lost them. He is oxidized. So if it helps you, you should already know this by now, but if it helps you to have a mnemonic device, here's one that's been used for a while. It's called oil rig. Oxidation is loss, reduction is gain. Now, the other thing you should know is the relationship with hydrogens. Think about hydrogen It's got one proton. Its atomic number is 1. It has no neutrons. It has 1 electron. So one hydrogen atom is actually a hydrogen proton and an electron. So you will see sometimes these terms used interchangeably because they're moving generally together. Okay, so let's go on. Alright, so everything I've just said is kind of mentioned right here. Again, look right there. Alright, so you can't have oxidation without reduction. If some molecule is going to give electrons away, something has got to gain the electrons. They don't just disappear into thin air. So you generally will see oxidation dash reduction, or you'll see them in the same sentence, or they just happen together. Now, you're going to be hearing a lot about electron carrier molecules. These are the middlemen that get the electrons and give them away. They are temporary electron carriers. You know them, the main two anyway, as NAD and FAD. I've put a molecule, not that you need to know it, of NAD down here at the bottom. This is actually NAD plus. Look at the thing I put a box around. I wanted to show you where the active site was, where the hydrogen would be picked up. So when 2 electrons, here are 2 electrons given to it, right here, NAD becomes reduced, and here is what that side chain will look like. Again, I've only put the area of the box right there. Also, hydrogen goes along with the electrons, but NAD can only hold 1 hydrogen. So again, if you look right here, there's that extra hydrogen right there. It only had 1 right there. Now it has 2. Okay. Again, 2 electrons, 2 hydrogens are passed, but since it can't really hold on, it can only take 1 hydrogen right there. The other one is just kind of free. So sometimes you see this written as NADH. That's this guy right here. Sometimes you see it written as NADH plus H, and H is that extra guy right there. Really, he's unimportant for us. We don't care about him. Okay, the other carrier is FAD, and FAD will pick up 2 electrons, 2 hydrogens. Those are the 2 major carriers. Okay, so let me remind you, we're going to make ATP by the process of phosphorylation, which obviously tells you phosphate's important here. There are 3 different ways that it can be done. It can be done by photophosphorylation, where light is involved. And of course that happens in photosynthesis, and we don't care about that. Obviously photosynthesis makes ATP. Alright, the main one we're going to be talking about, at least in the electron transport chain, is oxidative phosphorylation. This is going to involve the giving away of electrons. We'll talk more about it later. However, there is a third kind called substrate level phosphorylation, and in substrate level phosphorylation, you see an example of it over here. Look at this molecule right here, phosphoenol pyruvate, or sometimes called PEP, and look at that bond right there, or that what do we call it? Let me get rid of that. There, a tilde. That's what it's actually called on the typing area of your computer, and this indicates a high energy bond. Okay, there's your phosphate group, and that little tilde right there indicates high energy. So if we break that bond and we stick this phosphate onto ADP, which is a diphosphate, adenosine diphosphate, We've now created adenosine triphosphate. We've added an extra high energy bond. There's our ATP. What we have left of the PP is just a pyruvic acid or pyruvate molecule. That's substrate level phosphorylation. We're taking a molecule that has high energy because it has a high energy bond on it. We're going to break the high energy bond, take that high energy side chain, stick it onto ADP, make ATP. That's substrate level phosphorylation. It happens one time in the Krebs cycle, and it happens in glycolysis. But again, the main thing we're going to be looking at is oxidative, and here's an example of where it's going to occur. This is the electron transport chain. Okay. Reminder about enzymes. They speed up reactions. That's why they're called catalysts. They do it by lowering energy required to make the reaction go. Remember I said 5 minutes ago that chemical reactions really have to have some energy to get the molecules to interact with each other. It's kind of like yelling at your kid in the morning when you want them to get out of bed. They don't generally do that without somebody prompting them, an alarm, somebody yelling at them, something like that. Think of that as energy of activation. You see on the graph the energy, if you look at the solid line versus the dotted line. The dotted line is energy required, energy of activation, when you have an enzyme. Without an enzyme, it takes far more energy. Obviously, our chemical reactions in our bodies and bacterial cells require enzymes because the chemical reactions need to occur very quickly. That requires enzymatic input. You should know everything else on this list, so I'm not going to go over it. Okay, so what influences enzymatic activity? You already know this, pH. Remember that proteins are made, enzymes are proteins, and proteins have got an amine end of the molecule and an acid, carboxylic acid end or a carboxyl group end of the molecule. One end is acidic and one end is basic, and the solution pH can affect the function of the enzyme because it changes the charge on either side of the molecule. Temperature, the higher the temperature, the faster the enzymatic reaction will occur. Substrate, concentrations, enzyme, inhibitors. We're going to talk more about that in a minute. Okay, so here is a look on the diagram of just a simple reaction. The enzyme is the big blue guy, and we are using a substrate, fructose 1,6-biphosphate. It's got two phosphates on two carbons, carbon number 1 and carbon number 6. That's why it's called a biphosphate. It looks like a glucose. Glucose and fructose have the same chemical formula. Okay, so what we're going to do here is we're going to split this molecule into two molecules. You see that happening right here, and this requires the presence of this enzyme. Okay? And at the end of this, we get two little 3-carbon molecules that are produced during glycolysis, because this is happening during glycolysis. The enzyme now, this guy, is free to catalyze another reaction, the same reaction over and over and over again. And these are the things we wanted, the two end products right there. Okay, inhibitors, something that blocks this active site on an enzyme, can mess things up, obviously. Or anything that changes the conformation or the shape of this enzyme can mess things up. So there are different kinds of inhibitors. There are enzymes that block an active site. There are enzymes that can change the enzyme molecule shape. And then when that shape is changed, then the active site is no longer functional. Okay. So, inhibitors. Here's a look. I've just defined the two kinds of inhibitors for you, competitive versus non-competitive. Down here at the bottom, you see the active site. I'm going to put AS right there, where the substrate fits like a lock and key with an enzyme. And a competitive inhibitor will fit into that site, thereby occupying the active site so the substrate cannot fit there. That's a competitive inhibitor. Now, there are non-competitive inhibitors that change the shape of the molecule so the active site doesn't work anymore, doesn't fit with the substrate. And that's what I'm going to talk about now. And that gets me to feedback inhibition. And, nope, I'm too far. Let me take you through this picture on the right. Okay, so we have a substrate right here. And this substrate is acted on by an enzyme. There's an enzyme right there. And at the end of this chemical reaction, oops, wait a minute. I put my enzyme on the wrong thing. Wait a minute. I'm trying to get my, let me erase that. Okay. All right. My enzyme is located right there. All right, so this is the active site right there where this substrate fits. And when these two come together, we get, at the end of this chemical formula, this pathway, we get a end product right there. So when this substrate goes through however many chemical reactions, we end up with that right there. Now, let's say that the cell doesn't need any more end product. We can shut down, when I mean we, I mean the cell, can shut down this enzyme so that the substrate can't react with the enzyme. If that happens, then what would happen to the amount of end product? That would block the rest of this pathway, right? And the amount of end product would go down. So if the cell doesn't need any more of this end product, that would make sense. What you do is you block this first chemical reaction, and you don't get the rest of it. And then when the cell runs out of this end product, because remember it's not making any more, we unblock the enzyme, and it starts working with the substrate again. Okay, so look at the left side of this pathway. Let me eliminate, oops, got to get the bar. Okay, all that again. All right, so again, take a look at our enzyme right over here. If the cell has been making a bunch of this end product, there's going to be a large amount of it. The end product can bind onto this enzyme. There's actually a site, an end product binding site on the enzyme. So this enzyme, I'm going to draw it right there. It has an active site right there, I'll put AS, where it binds to the substrate, that guy. And it has another binding site for the end product. So this enzyme can change shapes. When it needs to, this active site is functional, but the end product binding site is not. On the other hand, the end product binding site can be active, but the active site where it binds to the substrate may not. So when the enzyme is bound to this end product right there, that closes off the active site, and the active site can in no way react with the substrate, and you don't get any of these reactions that you see on the right. So the end product is changing the shape of the enzyme so the enzyme cannot react with the substrate, bringing the whole metabolic pathway to a halt. Now, presumably this end product is being used by the cell, right? So when there's not enough end product anymore, okay, no end product anymore because it's been using it, then that means that there's not enough end product to block that, to bind to the enzyme site. And now the enzyme changes its shape again, and the active site now becomes functional, and it starts reacting with the substrate again, and you get the pathway. This is called feedback inhibition. The end product is feeding back to the first pathway, the first chemical reaction, blocking the enzyme function. I'll give you better information in lecture. Okay, here's a good example of a competitive inhibitor having to do with antibiotics. So let me get my drawing tools here. Here is your enzyme right there. Here is the active site right here. This is the chemical reaction that we're talking about. It involves the substrate, para-aminobenzoic acid, which you can see part of right here. In the end, the result of this pathway is the production of a molecule called folic acid. Folic acid. And folic acid is needed to make RNA and DNA, as you can see here. Now, the enzyme has an active site for para-aminobenzoic acid right there, and at the end of the pathway, there's our folic acid right there. However, look at these other molecules that you see right here. These are 3 variations of a group of chemosynthetic drugs used in chemotherapy for infectious disease called the sulfa drugs, not sulfur, sulfa, S-U-L-F-A. Notice that the sulfa drug molecules have a similar molecular structure to PABA. Okay, here, here, here. So these, if any of these molecules are present, they serve as molecular decoys for PABA. In other words, the cell will pick up the sulfa drug erroneously instead of PABA, which is what you see happening here. So this is one of the sulfa drugs up here. And that means because it's occupying the active site, PABA cannot attach. Therefore, you don't get any folic acid, and therefore the cell cannot effectively grow, and therefore it dies. That's an example of competitive inhibition. Okay, so let me lay out the setup here. Here's the big picture. We're going to start with carbohydrate catabolism. That's the one you know. It's oxidation of glucose. Aerobic respiration is what we're going to start with. Okay, so what you see here is a look at the breakdown of aerobic respiration or cell respiration. On the left, the 3 parts. 1, glycolysis. 2, Krebs cycle, or also called the citric acid cycle or the TCA cycle. And 3, down here, is the electron transport chain. 1, 2, and 3 constitute cell respiration. So, in a nutshell, we start with glucose. We are going to go through about 9 or so chemical reactions in glycolysis. We end up with, and by the way, I'm making this really simple. We're going to get into it in more detail. We end up with pyruvic acid. In fact, we end up with 2 pyruvic acids from 1 glucose because we're going to split a 6-carbon molecule of glucose into 2 3-carbon molecules. Pyruvic acid is 3 carbons. Now, if there is oxygen present, and if the cell has the enzymes to continue on in aerobic respiration, The next step we go into is the Krebs cycle. And in the Krebs cycle, we have a cyclical set of chemical reactions, maybe requiring about 11 or 12 chemical reactions. It's called a cyclical set of reactions, a cycle, a metabolic cycle, whereas glycolysis is linear. One cycle causes another cycle, kind of like dominoes knocking each other down. In a cyclical metabolic pathway, it goes around, and the last molecule right here in the cycle starts the whole cycle again. Okay, so that's a Krebs cycle. And the main thing we want out of the Krebs cycle are these reduced electron carriers, NAD, H, and FADH2. Now, these now are involved in the electron transport chain, which we'll talk more about. And the result is the production of a bunch of ATP. That's aerobic respiration, or cell respiration. Now, let's go to the right. If there is not enough oxygen, or if the cell does not have the enzymes needed for glycolysis and the Krebs cycle, I'm not talking about your cells, but there are a lot of bacteria that just don't have these enzymes, then it's got to go an alternative path. It can't go aerobic respiration. It's got to go an anaerobic route called fermentation. And as a result of that, which is a very short little pathway, we end up with, lactic acid, or we end up with alcohol. We'll talk about that. We get ATP out of this pathway. Most of the ATP is made actually during glycolysis, but we get far less ATP made going this anaerobic route than the aerobic route. So it's a no-brainer. A cell would always choose the aerobic route if, A, it's got oxygen or some other electron carrier it can use. We'll talk about what those could be. And B, it's got to have the enzymes to do these chemical reactions. Okay. So again, to lay this out a different way, and then I'll stop and we'll actually go through the rest of it on the next video. Here's the overall formula for aerobic respiration. Here is glucose on the left, oxygen. By the way, these guys do not actually interact. In fact, oxygen is not even used until the electron transport chain, at the end of the electron transport chain. So here are our two reactants. At the end of all three parts, glycolysis, Krebs cycle, electron transport chain, we get ATP, water, CO2. Okay, so what has happened is glucose has been oxidized to CO2. We're going to strip off these hydrogens and electrons. Glucose is giving them away. It becomes oxidized. Now, if it becomes oxidized, something's got to be reduced. Obviously, oxygen is going to be reduced. It gets reduced to water. Note that we have stuck hydrogens on it. The oxygen has picked up hydrogens, picked up electrons, and become reduced. Again, here are your three parts. You've already studied this in general biology, having to do with eukaryotic cells. We're going to look at this as it deals with prokaryotic cells more so. But really, it's pretty much the same. But one major difference, and that is you've always heard of this related to mitochondria. And remember, bacteria don't have mitochondria. So in bacterial cells, here's a bacterial cell right there. And here is its cell membrane inside of the cell wall. Okay, cell membrane is the inner blue line. This process of Krebs cycle and the electron transport chain is occurring right here. Because there are no mitochondria, you got to have a membrane for the electron transport chain to occur. And bacteria have no mitochondrial structures, which have the membranes where this is occurring on your own cells. But frankly, it's pretty much the same. By the way, one more thing I want to mention, although I'm going to mention it in more detail later. It's now understood that really there's not 36 to 38 ATP. We'll talk about why later, but it's now been found to be less efficient. And most books still have it maybe as 36 or 38, but it's actually thought to be somewhere around 30, 32 that are produced. But that's a fine point. Okay, so I'm going to stop here and we'll get into the meat of this on the next